and breeding at this conference. And thanks for including forestry in the SciBreed initiative. Very much appreciated. And I say unabashedly and without shame that we can use all the help we could get, because we have monumental challenges that we face. And we do our best, relatively small community, in accessing as much information and models as possible from model systems and from crop and animal breeding. But I think a setting like this that you're establishing, where everyone is working together, that will really facilitate that. So I really appreciated the talks so far, and especially this morning, the two new faculty members. Their research on modeling is so applicable to what we're doing and will be so helpful in mining the data that we're generating that we haven't been able to do entirely effectively yet, I'll say. So today I would like to just give an overview of how tree improvement differs from normal crop and livestock breeding, what our objectives are, what our challenges are, I think what opportunities we present, some things of interest for SciBreed in moving forward, and what the roles of genomics and molecular genetics are, and some examples of hardwood tree improvement genomics projects that I and Oliver have been engaged in together, and which may represent some of the work that Kostya and Ludger and Oliver are, and others in the tree genetics, forest tree genetics and breeding department here are engaged in. So first, a little advertisement for the Schatz Center to explain what it is, and to show that we have many opportunities for collaboration with the SciBreed Institute here, I hope. So the Schatz Center was established with a gift from an alumnus who graduated in forest management at Penn State in 1934, but made a lot of money in the plastics industry. What he got his PhD in. And we have endowments for tree genetics colloquia, for undergraduate research projects, for undergraduate field trips. And we should go every 2 years to the Schatz Tree Research Farm in Humboldt County, California, and for international scholars and for visits for half a year to a couple of years at a time to initiate research projects on their areas of interest. So as the chair mentioned, we have research projects ongoing in areas of conservation genetics, genomics, and in biotechnology. So some of the things that we have been engaged in are developing new genomes for threatened tree species, a lot of population genetics in the past, not only with trees themselves, but also with pests and now with microorganisms that interact with trees. So what is tree improvement? Well, tree breeders aspire to have recurrent selection programs, just as you have it with crop genetics and with animal genetics. The time frame is a little bit different, though. When we have 15 to 20 year juvenility periods, and we have 100 to 200 to 300, sometimes 1,000 year generation times. So the scale is quite different, and we therefore tend to be self-selected for people who are patient and who are looking forward to the future. But I think SciBreed is very much in the present, but also looking forward to the future too. And I say recurrent selection is our what we aspire to. We have only, for most species, 2 or 3 generations of selection so far. But it's always, for all our breeding programs are always oriented towards maintaining and fostering genetic diversity, which is why I'm in this section now. As stated by some of our important forest tree breeders in the past, Floyd Bridgewater here, Texas Forest Service, and Texas A&M University, primary reason for tree improvement is to produce genetically improved seedlings, of course, but also to maintain adaptation
to the sites where they're going to be replanted in. Otherwise, you have failed your experiment to start with. Charles Maynard from SUNY ESF in Syracuse, New York, who has been a leader not only in tree improvement but also in biotechnology for forest tree species, states in a little bit differently that tree improvement objectives should be to manage sustainably genetic variation so that the adaptive potential of the tree populations is maintained for the long term. We don't want to disrupt that. And as Bach-Romeda said here at the very early stages of tree improvement back in 1959, one must maintain the diverse forms and characteristics of the entire population. So for tree improvement, we're really talking about shifting the means of large populations, while at the same time maintaining genetic variation. And that's done by identifying our forms, the ideal forms for high productivity and high wood quality and the like, and selecting on those phenotypes, but at the same time looking at maintaining diversity. And that's done primarily through progeny tests, selecting trees that are as close as possible phenotypically to those ideotypes that we're so much looking forward to being able to select for, and then establishing seed orchards from cuttings of those elite or high phenotype trees, collecting seed from those trees in natural stands, doing progeny testing, and then going back and removing the genetically poor individuals from our seed orchards to improve the mean of the seed that's being produced from those orchards. So here's an example of an ideotype for Scots pine and Norway spruce and Sitka spruce from British Columbia and from Northern Europe. I have this idealized type of tree that is very tall. It can be planted at a high density like hybrid maize and still have the form that we want and the wood quality and the volume of high quality wood. And the first couple generations of selection in tree improvement programs was really focused on achieving this ideotype while maintaining genetic diversity. But very quickly, within the first 20 years of tree improvement programs in the 1960s, the big problem became tree insect pests and forest diseases and abiotic stresses from air pollution, from acid rain and the like. And our forest genetics conferences started shifting from volume production to maintaining populations in the face of all these abiotic and biotic stresses. So, a little bit about progeny testing means for trees. So, collected seed from uncontrolled open pollination of female trees that have the ideal phenotypes. We plant those out. We grow the seedlings in nurseries in uniform conditions for a couple of years so that they all have a good start. We outplant them at multiple field sites in randomly designed plots within each of those replications, within each of those field sites, and then we look for how they perform over different areas within the region that they're suited for. Then we measure growth rates over at least 5 to 10 years, ideally 20 years, because you need to get past 10 years to transition from juvenile characteristics to mature characteristics in the trees. So that's a very long time frame to be sure. But the results have been very successful. So the first couple generations have each improved productivity and form and quality wood by over 25%. So that's starting from completely undomesticated, unselected populations and moving forward with slight degrees of selection. But there are big opportunities for gain here. And also now we want to stress big opportunities for further gains from genomics, much more so than with the slight gains that we might be experiencing in hybrid maize going forward through the use of genomics.
And another accomplishment was that genetic variation was maintained or perhaps even improved by bringing together different genotypes that weren't in the same natural stands to begin with, to further increase the variation that's there for adaptation in the future. So we can say the tree improvement has been successful, but it hasn't moved very far forward because of the time frame so far. And we think that having marker-assisted selection, genomics approaches will help us to accelerate breeding programs. And that's been one of our reasons for putting so much effort into genomics and molecular genetics and conservation genetics with forest tree species, even though the generations are so long and there are many issues to deal with. The main issue now across the world, however, as I mentioned, is what we generally lump together as to call forest health issues. Issues with big losses to productivity in both natural stands and in highly managed clonal propagations, plantations alike. So here are some of the threats to sustainability that we're currently experiencing in our forest. Major insect infestations, largely exotic insects being transported through trade and being free to attack trees of their favorite species, without any resistance usually being in those natural populations, because those populations never saw that particular insect threat or disease or exotic disease before. So these infestations are swept through very large geographic regions, removing pretty much all of the individuals for those species. And the numbers of infestations, the number of pests is increasing over time rather than decreasing. This is all exacerbated by climate change and other abiotic stresses that weaken the trees to begin with, make them more susceptible to exotic diseases, as well as endemic insect pests and diseases. And we have a lot of forest fragmentation that is occurring because of development. Like in my state, in Pennsylvania, we have a lot of fracking wells for natural gas now that have completely fragmented our forests that were entirely continuous up until 10 years ago. So there are not only many different pests and diseases that are issues now, but they are affecting pretty much every genus of forest tree species that you can think of that's important on the landscape and important economically. So this is our major challenge, is that we cannot make much inroads by only focusing on one species. We have to keep in mind that our populations, whether managed or unmanaged, are multi-trophic and have multi-species and lots of interactions going on. How can we capture what's going on at the genome level to be able to facilitate improving those and maintaining those populations and resilience to future changes or threats that we don't really know about. So some of the initial major losses to forest productivity that have occurred in the United States, eastern United States where I live, as well as in Germany and the region here where it's from air pollution. Ozone wiped out many high elevation populations of hardwood trees in eastern North America and the Appalachians in the 1970s. And then acid rain came in the 1980s in a big way and had major impacts upon industries like sugar maple industry, because the sugar maple trees are susceptible to acid rain and the heavy metals that acid rain releases in forest soils. But also we have now increasing long periods of drought that are having particularly major impacts in the western U.S., where we have a lot of conifer tree species, and climate change, of course, is impacting that. So one current example of an effect of climate change is here we have billions of trees over tens of millions of acres that have been killed of conifer species caused by an epidemic of the bark beetle that has always been in North America. It's native to the western U.S., but now because of a 2-degree change in the average winter temperatures for over the last 15 years or so, the population is maintained over the winter. It isn't suppressed to small numbers because of cold temperatures in the winter. And now this insect pest is out of control. So there are many traditional approaches in forestry to be able to deal with these threats and issues, and they include thinning, where you identify a tree that's been attacked to cut it down as soon as possible. That hasn't much worked in terms of controlling spread. Chemical spraying can work in your garden very well. There are many systemic insecticides and fungicides that work on a small scale, but not on a large scale. They're too expensive and too environmentally threatening. Other things like trapping are good for monitoring, but not good for treatment. And one thing that's been very popular for the last 100 years has been trying to remove forests in advance, remove species from the forest in advance of the threats that are coming. And that also hasn't worked very well because people move the pests around in firewood, regardless of the attempts of the Forest Services to control it. But breeding for resistance has been ongoing from the 60s, but we need help to be able to accelerate that. So we hope that genomics approaches will aid in both identifying which trees to keep and which trees to cut in advance of epidemics, but also in accelerating breeding through both not only structural genomics, like identifying QTLs to develop DNA markers for selection within seed orchards and natural populations, but also through identifying what the important aspects of variation are in terms of adaptation in our population, so that we maintain those adaptations, those co-adapted allele complexes, rather than destroying them in the process of selecting for insect resistance or disease resistance. And tapping into evolutionary genomics approaches like Bruce was talking about and others at the meeting to maximize our knowledge about what's going on and what has happened in the past, and translate that into tools that we can use in the future. So challenges, again, for forest resilience. We are not dealing with one species, we're dealing with populations of multiple species, and to maintain the productivity of a forest, we need to address what's happening in multiple species. We have multiple stresses that are occurring at the same time. Abiotic stresses like droughts from climate change. At the same time, we have insect pests coming in and diseases coming in. We're not dealing with farm fields. We're dealing with heterogeneous, very large populations over large areas. In many cases, there's no known natural resistance to these exotic pests and diseases. And we don't have a lot of time to try traditional approaches because the pest moves through more quickly than our selective logging we might be able to do. And of course, as I mentioned several times, we have to maintain genetic diversity while doing this. And I should also stress that most of our forest geneticists who started the field in the 1950s and 1960s have retired now and have not been replaced. So we feel very bad about that, and we feel very good about an initiative like SciBreed, which emphasizes breeding, not just the genetics aspects. So we have great opportunities, I think, for moving forward with forest tree species as well. We have large natural populations that I think represent nature's experiments in adaptation, and the information is out there if we have help, from Tim and others in developing models for assessing what regions of the genome have been selected for that are important for these adaptations. So any stand like this, in Pennsylvania, has been adapted to grow on very poor sites. Our hills are just basically piles of rocks, and the trees do very well there. As these tree species came in after the last ice age receded 10,000 years ago, each of the populations was selected for when to become dormant in the fall and when to flush their buds in the spring. And that has very, very high heritabilities that can be selected for but have to be maintained, or we have the threat of losing trees from frost damage and the like. But that variation's out there for us to look at. This particular population has only existed for 100 years, even though its progenitor population was around for at least 8,000 years, because all the East Coast was clear-cut 100 years ago. But this, again, is another experiment. An experiment in what the seeds and seedlings that were left, what selection pressure they went through. And there must be signatures of adaptation and selection that have occurred in each of the species. And this is also represents dozen or so forest tree species, from conifers to asterids to ancient magnoliids up to more recently evolved rosaceae species. So a lot of experiments going on here at the same time that we could tap into. So here's one example of our work with an invasive disease. It's a beech bark disease on an American beech. It was imported from Europe in around 1932 into Maine. This is a complex. It's an insect pest that carries one of two different nectaria fungi that then, after the insect damages the tree, then infect the tree and kill the tree. And it is spreading relatively slowly compared to chestnut diseases and some, like green ash infestation by insects, but it's still spreading throughout the range. So, fortunately, the U.S. Forest Service and state forestry bureaus have, in this case, have not tried to liquidate the forests in advance or the species in advance of the infestation, but have kept track of where there may be trees that have survived. And there has been a very low percentage, much less than 1%, very low, very minor allele frequency, if you will, of individuals that have survived, and which the Forest Service, Jennifer Cook and others in the Forest Service have gone out, collected scions from, brought them into the greenhouse, and tested them through bioassays to confirm that they were actually resistant, and have developed seed orchards for them. So we asked the question, could we find candidate genes for resistance through a GWAS study that looked at 250 or so beech bark disease resistant individuals, and then randomly selected same number of susceptible trees. So we had a reference transcriptome that was actually kind of oriented towards stress-resistant stress resistance and disease resistance experiments, but we still have a reference genome to use for this species. But we were able to develop this high-density linkage map based upon the 3,000 or so SNP markers, and identify one locus that was statistically significant in terms of association with the insect and disease resistance trait. And that was located on one position on our linkage group 5, and it was for one gene, for a gene metallothionein-like protein that had actually been discovered in European oak as a gene that's highly expressed in bark. Not European oak, European beech that's highly expressed in bark. So it could be a constitutive gene for insect resistance. And it's probably involved in moderating oxidative stress responses in the tree after it's mechanically attacked. We still have a lot of validations. That's one of the shortcut, or the issues or problems with taking these approaches with trees, is then we have to go back and do long-term experiments to actually validate that. And we're in the process of doing this, or David Carey is in Jennifer Cook's lab in the U.S. Forest Service in Ohio, by establishing new QTL populations and identifying where the QTLs are. He's found 3 QTLs already in an early phase of the new mapping population, one linkage group 4, by his numbering, which is the same locus as we found by GWAS. This is ongoing, and of course the whole idea is to be able to apply this in terms of testing for advanced selection in natural populations, which still remains to be done. The chestnut blight was the first major ecological epidemic that occurred in the United States, and also here in Europe, which is caused by the Cryphonectria parasitica, which has attacked our natural populations of chestnut, which were our dominant tree species in high elevations in the east, in these very large diameter and fairly tall trees that were the mainstay of the economy of Appalachia. And the blight came through, it killed all of the tops of trees throughout the entire range, and we still have sprouts coming up from the roots. that can be used for breeding if they get old enough to be able to produce pollen, but we don't have any large trees anymore. Early on, around 1920, not long after the blight started to progress out of New England, some USDA geneticists did crosses between Chinese chestnut, which is resistant to the blight, and American chestnut, and their hybrids, and then they looked at individuals from their hybrids, but that didn't progress very far until 1983, when a charity public foundation, American Chestnut Foundation, was established to try to make use of those hybrids to progress towards restoration of American chestnut. And this was all based upon the ideas of a well-known maize geneticist, Charles Burnham, who together with Ron Phillips developed a backcross breeding strategy that the Chestnut Foundation took to heart and has been employing and working through ever since. And the idea was to go through at least 3 backcross generations with the hybrids from the USDA and intercrosses in between to look for the segregation of the phenotypes for resistance as well as to recover the American genome. And the prediction was that we should be able to get through this with 7 generations, including the intercrosses. How long would this take with a long generation tree improvement program wasn't known, but fortunately some techniques were developed for early flowering, for advancing breeding a bit more quickly, and we are actually at this stage already, the Backcross 3 F3 generation, but we haven't accomplished resistance yet, and nor have we accomplished recovering the American chestnut phenotype. We still have an intermediate phenotype. Why is this? Well, we knew from studies at this generation that in a QTL mapping populations, 3 major QTLs at least were discovered for this trait. So it's not a single gene trait, it's a polygenic trait. And even after 5 generations, we haven't resorted all of those into individuals that have all of the QTLs. And that's because I won't say tree geneticists, I'll say pathologists who were responsible for this breeding program selected only on phenotype for resistance and not on tree phenotype and not using the DNA markers that were discovered and published in the 1970s for the 3 QTLs. Unfortunately, but now we're trying to hop in to at this end to have genome-guided selections to finish the work. So fortunately, back in 2010, the Forest Health Initiative funded our lab to develop a reference genome for Chinese chestnut and to discover all the genes in the QTLs for blight resistance. And we, at the time, the only approach we had was to use 454 and Illumina sequencing and a de novo assembly, but our goal has always been to have chromosome length sequences to enable, really genome-wide or chromosome-wide selection. This is the genome that we've published in 2014, based upon the de novo assembly. 40,000 scaffolds, 36,000 gene models, and a very good understanding of what the gene content is on those 3 QTLs. They're very large QTLs, and represent a lot of genes, and including even a lot of genes that are responsible for stress response. So we don't have individual candidate genes to work with yet, but we have regions of the genome to focus in on. And this, the browsers for the QTLs, browsers for the whole genome are available, and all the data is available for download and has been used in quite a few interesting studies to date. One of our studies was to look at using this genome de novo assembly to see how far along we are, in this process. And the Backcross 3F3 individual is kind of still intermediate in terms of SNP variation between the parents, the Chinese and the American parents. So we still have ways to go. We've had much better luck using recently the chromatin interaction approach to generating data across chromosomes for pulling together scaffolds, and so with a different genotype we now have chromosome length sequences that still need to be characterized but can be more readily incorporated into selection. Well, I have a lot more that I wanted to talk about. One thing that I worked on with Oliver when he was at Michigan Tech University for several years, was developing transcriptome resources for 8 the most economically and ecologically important hardwood tree species. And we accomplished this. We selected individuals based upon having the greatest phylogenetic coverage possible, so that if, in fact, we would ever have reference genomes for these species, they could be translated into and used for other species that were closely related in these different taxonomic orders. And we focused on abiotic stresses. So we did RNA isolations from stressed individuals, and we generated over 240 gigabases of sequence data, which is available at our Hardwood Genomics website for each of these species. It's been assembled, it's been characterized in terms of protein content, and it's being used by thousands. It's been downloaded by thousands of times by people around the world, so hopefully it's being put to very good use. Just wanted to end on pushing or mentioning that our next phase that pretty much all molecular geneticists working on trees are now moving into is landscape genomics. So we have these resources that have been available for a long time, from the initial generation of forest genetics, from provenance trials looking at phenotype by environment associations. We are getting close with genome resources to being able to look at genotype to phenotype associations. But now we want to look at what were the adaptations that have occurred in natural forests in the past. And so that involves a landscape genomics approach of genotype by environment associations. And several of our large tree genome groups are already engaged in this as a way of looking at and learning about what adaptations have occurred. And one final slide. I think that the opportunity exists here for SciBreed to go beyond looking at how plants and animals information can be shared, but to go beyond that to look at a more holistic systems-wide approach to doing breeding, and to, for instance, including, as must be showed here, the importance of evaluating the crop microbiome in the soil and above ground, and how that interacts with the genotypes of the plants that are being grown. And we have one initial study that we did in the lab on shrub willow. These are a plantation grown more like a crop plant, but not that well domesticated. But we tested a lot of cultivars, and Wan Yan looked at the differences in the soil microbiome between 24 cultivars at 3 different locations that very much differed from location to location in terms of height, growth, and yield. And she found that, in fact, there was a major effect of site on the microbiome, but also an effect of the genotype. So differences in soil microbiomes between genotypes, and nevertheless, within the species, there's a kind of a core functional assembly of microorganisms that the willow plants extract from the soil, from whatever is available for them in the soil. And so I wanted to thank the many people whom I've worked with over the last 30 years on projects like this, as well as our funding agencies, and answer any questions you might have, if there's any time left.
Thank you very much, Professor Carlson, for this comprehensive introduction in the world of forestry breeding. We understand that it's quite different from other plants. Other questions? Short questions? Yes.
Yes. So there's a European project for assessing variation in resistance, and also getting to the mechanism of resistance. So I've worked with Richard Buggs in the UK. He's now at Kew Gardens, and he has developed a genome for European ash and transcriptomes, and we're working in parallel on green ash, even though it's an insect. In our case, that's decimating that species. There's a lot of information we're able to share. So there is a program in Europe on finding variation and seeing what can be done to advance that through breeding, at least through setting up seed orchards.
Okay, I think we don't have time for much more discussion, so again, thank you very much.
Thanks.